Dear students, so we'll continue the next lecture of physiology, uh, excretion with counter current multiplier exchange theory. So lecture number 17 will include the excretion and counter current multiplier exchange theory. Actually, we have studied these topics at the basic level in the previous work, but we will repeat something here for the better understanding. That we have seen last time, the blood is a liquid connective tissue. It flows through the channels. It has uh, the composition in which plasma and some formed elements. So in plasma, these formed elements includes the solid components, and inorganic components of the plasma includes major electrolytes like sodium, chloride, potassium, ions, etc. And then salts of sodium chloride. We have already seen in previous discussion. So here serum albumin is also there. So these are the materials which are the basic components of the blood and regulate all physiological activities. And then the primary purpose of the albumin is to transport the fatty acid materials or the hormones, enzymes likewise, which are not terribly soluble, but they also in aqueous environment or water material is also there. Then albumin serves to transport the lipid soluble hormones and steroids. Then another major component is the carbonic anhydrase, which catalyzes the conversion of dissolved carbon dioxide to carbon bicarbonate anions and back. So this reaction is reversible. Then carbonic anhydrase is very essential material toward the maintenance of pH of the blood, then extracellular fluid and its physiological limits. So this regulate the physiology. Then on the other hand, plasma proteins of interest include immunoglobulins, fibrinogen and some clotting factors. So this one is about the blood plasma and formed elements. Now the serum also is a liquid which consists of some clotting materials like fibrinogens, then clotting factors, then some electrolytes, proteins, and also it includes some antibodies. So this one we have already studied in our previous. Then these formed elements or these plasma materials, uh, how do they work properly as a blood element? What is their role of blood cells? What they do in the physiology for maintaining the survival of the animal? We will study one by one. We have seen the plasma and the blood contents include the formed elements. So these formed elements are all cells or parts of the cells aside from the platelets. That is, we can say plasma, then RBC, WBC, etc. The red blood cells is the most abundant of the formed elements. Means in formed elements, RBC and WBC are the main parts. Of them, RBC are tremendously present, more than about 5 times more about WBC, and for good reason. They maintain the very best physiological activities. Blood performs a remarkable number of functions, including maintenance of extracellular pH and electrolyte balance. Means the main functioning of this including the main pH maintenance and water and salt balance of the body, electrolyte balance generally, or sugar elements and water. Then the delivery of fatty acids and other nutrients to the tissues, then delivery of toxic compounds and waste products to the liver and kidney, and it also serves as primary vehicle for hormone delivery and immune responses. So blood passes through the body, supplies the hormones and water and electrolytes, etc., and simultaneously also supply the delivery of some immune responses and some hormones also. So above all, the primary immediate moment to moment function of the blood is to deliver the oxygen to the tissues, because oxygen is the immense need of the body, immediate need of the body, and therefore this continuous supply is being given there by the body to the all tissues. Then in spite of these formed elements, that is red blood cells and others, it also continue or it also consists of some platelets. So platelets are the materials which are present in spite of these RBC and WBC, or actually we can say whenever the life of RBC is completed, it is broken down and its pieces works together as a formed elements and helps in clotting of blood activity. So these platelets are the fragments of larger cells that are ruptured before entering the bloodstream. They provide the bulk of material for formation of blood clots. So they help in clotting of blood. They stop the blood to flow outside the body. And white blood cells, or we can say WBC, or generally we call it as leukocytes, are mediate immune responses and are readily seen in right stained blood stream or blood smears, although they are much less abundant than the red blood cells, because red blood cells are tremendous and abundantly found in the blood samples, and WBC are very rare or less from the healthy individuals. So this is about the composition of blood, working of the blood, role of the blood cells and platelets. The mature mammalian red blood cells has no nucleus. They are non-nuclear or cytoplasmic organelles and very little enzymatic activity. It is designed for a specific purpose, that is to enclose a large quantity of the oxygen binding proteins. Hemoglobin in package that allows travel through tortuous routes in the circulation and efficient gas exchange at the same time, means they can flow even in a very thin capillary, one by one behind another, but simultaneously they also perform the function of exchange of gases or fluid material, and therefore they are of prime importance in the circulatory system. So next topic is hemostasis. Hemostasis is a process to prevent and stop bleeding. Hemorrhage is a continuous flow of bleeding, and hemostasis is to prevent or to stop the bleeding from the body. Suppose accidentally there is a injury to the body, then at start the blood flows fastly, and then this flowing of blood slows down later on. It means that our body have hemostasis mechanism that stops the bleeding by way of making clot or by making barriers by fibrinogen materials, and likewise the bleeding at last forming a clot and completely stop the blood bleeding. So in hemostasis, meaning to keep the blood within a damaged blood vessel. The opposite word of hemostasis is hemorrhage. I told you hemorrhage means continuous flow of blood, and hemostasis means stop the flow of blood in the body. So it prevents the excess loss of blood, and hemorrhage is nothing but the loss of blood. It is the first stage of wound healing. Hemostasis is the first step of wound healing. Whenever the Injury is there, blood is flowing, it forms a clot and it stops the blood and the wound is completely heals. But in wound healing, there are some steps, 8 to 10 steps. Of them, first is the hemostasis. When a blood vessel is injured, platelets like stick together to form a plug so that the blood flow is completely stopped. Second thing, proteins called as clotting factors interact to form a fibrin material and forming a mesh or network to hold the platelets in place. They are not allowing the platelets to flow outside the body. So platelets stop the bleeding at the place of injury, and this allows the injury to heal while preventing blood from escaping the blood vessel. In this way, the healing or stopping the bleeding is nothing but the hemostasis, and later on it converts into the blood clotting or blood clot formation. Generally, control of bleeding is achieved by very quickly through the formation of a clot. This clot is very important material which stops the further loss of blood from the body through the injury. In major trauma or surgery, physicians often need to help patient to achieve adequate hemostasis in order to minimize the blood loss and related injury. So during operation, the maximum blood is flows from the body, and to prevent this, to prevent the loss of blood of the patient, so doctors take care that how the hemostasis can be applied to minimize the excess loss of the blood. However, some peoples are born with a bleeding disorder. This is a genetic disease. Some peoples have a bleeding disorder, we can say congenital, that impairs their ability to achieve hemostasis. Some peoples achieve hemostasis very earlier, instantly, on some takes more time, and therefore this is a disorder. Example is the hereditary disorder called as hemophilia. Hemophilia is the example of disorder in which these people did not show the fast clotting of blood and the hemorrhage is continuous in such peoples. As well, some peoples who have never had any bleeding problems can develop a condition that causes them to bleed, known as acquired hemophilia. So this one is the opposite, or we can say acquired hemophilia mechanism, and such changes are naturally occurs. These are the birth, we can say diseases, or we can say traditional diseases may come from the parents, but show such a different activities of clotting. What are the key lines? What are the basics of the hemostasis? Hemostasis is the natural process that stops the blood loss when an injury occurs to the body. It involves the three steps: vascular spasm, we will repeat this word. It involves the hemostasis involves basically three steps. First is vascular spasm, or we can say vasoconstriction. So the cells get constrict so that excess blood is stopped, or we can say minimize the excess flow of blood. Second, platelet plug. All platelets flow is stopped by the fibrin material so that the platelet form a plug and to protect the body from the excess flow of blood. And finally, this third is the coagulation. Means fibrinogen, magnesium, all these materials come together and forming a clot. आपण त्याला रक्ताची गुठळी म्हणतो जनरली, किंवा रक्त गोठण्याची प्रक्रिया म्हणतो. त्या प्रक्रियेमध्ये या तीन गोष्टी समाविष्ट आहेत. तर आधी पेशींचे आकुंचन पावणे, नंतर प्लेटलेटच्या प्रवाहाबाहेर जाताना धाग्यासारख्या फायब्रिनने थांबवणे, आणि सगळं थांबल्यानंतर कोरडं कडक होऊन ती जखमीवरची खपली बाहेर फेकली जाणे, ती कोॲग्युलेशनमुळे होते. आणि मग अशा पद्धतीने वुंड हिलिंग पण होते आणि एक्सेस ब्लड बाहेर जाण्याचं स्टॉप पण केलं जातं. सो दीज आर द की लाईन्स ऑफ हिमोस्टॅसिस. दुसरी गोष्ट आहे समथिंग इज अबाउट व्हॅजोकन्स्ट्रिक्शन. Vasoconstriction is a reflex in which blood vessels narrow to increase the blood pressure. म्हणजे जास्तीत जास्त ब्लड वाया न जाता, लॉस न होता जखमीतून, त्याला वाचवण्यासाठी व्हॅजोकन्स्ट्रिक्शन ही नैसर्गिक प्रक्रिया बॉडीमध्ये तयार केलेली आहे किंवा तयार असते. नेक्स्ट, प्लेटलेट प्लग फॉर्मेशन इन्व्हॉल्व्स द ॲक्टिवेशन, ॲग्रेगेशन and adherence of platelets into a plug that serves as barrier, म्हणजे अडथळा निर्माण करतात, अगेन्स्ट ब्लड फ्लो. जखमेतून रक्त बाहेर वाहून जाण्यासाठी अडथळा निर्माण करण्यासाठी फायब्रिन्स आणि प्लेटलेट एकत्र येऊन असा एक प्लग निर्माण करतात की त्या प्लगमुळे ब्लड पुढे सरकत नाही, आणि त्यामुळे तो शेवटी क्लॉट तयार होतो, गुठळी तयार होते आणि रक्त थांबते. त्याला क्लॉटिंगसुद्धा म्हणतात नॅचरली. पुढे शब्द आला बघा, कोॲग्युलेशन म्हणजे क्लॉटिंग. कोॲग्युलेशन इज द प्रोसेस इन विच द ब्लड इज बिकम स्ट्रॉंग or its molecules come together and forming plug to prevent the excess flow of blood from the wound. The method is called as coagulation. It involves a complex cascade in which a fibrin mesh is cleaved from the fibrinogen, and the fibrin, which is formed from the fibrinogen, acts as a molecule or molecular glue. You can say this is one of the molecular glue like material. It sticks there to prevent the excess flow outside the blood. And during clot formation, this is a very important role because coagulation causes this clot formation. During clot formation, holding the platelets plug together, and all these plugs are combining and forming the excess loss of blood. So we will see the further method of coagulation and hemostasis. Because hemostasis is the starting point and clot formation is the end point, this is the method of stopping the blood from the wound or excess loss from the body. So this one is the schematic or diagrammatic presentation that whenever the injury is caused to the body, what happens? The blood releases from the body, the tissue get damages, and some materials, RBCs, are also disturbed. In such a condition, injury condition, what happens? Our body causes vasoconstriction. The tube becomes shrink. Then the collagen material, which is activate the platelets to form a plug to prevent the excess flow. And these are the this is called as platelet plug. This platelet plug is nothing but the primary hemostasis, or we can say starting of hemostasis, because it helps the factor fibrinogen to the platelets plug, and that plug is preventing the excess. So fibrinogen and the factors which create it, which stops the blood flow, are the platelets and its activation in presence of collagen, and primary hemostasis occurs. Then they communicate with the coagulation cascade mechanism. When the tissues factor released, then it forms a thrombin, or we can say from the using thromboplastin material, Then it involves in the reaction where fibrinogen is formed or converted into fibrin, because platelet plugs facilitate this conversion of fibrinogen into fibrin, and once the fibrin is formed, तंतू जर का तयार झाला, तर that fibrin is helping to maintain, to regulate, to prevent the flow of blood, and the blood clot is formed. रक्ताची गुठळी तयार होते. And from this a plasmin is there which acts over there to the blood clot, and fibrinolysis and clot degradation occurs. अशा पद्धतीने हा क्लॉट सुकतो आणि जखमेची बारीक काळी खपली आपल्याला दिसते, ती आपण कुरतडून फेकून देतो, आणि अशा पद्धतीने खाली जखम पण भरली गेली असते, वूंड हिलिंग पण होते, एक्सेस ब्लड प्रिव्हेंट पण होतो, आणि टिश्यू डॅमेज पण थांबतो. या तिघ गोष्टी एका वेळेला होतात हिमोस्टॅसिसमध्ये. तर याला म्हणायचं सेकंडरी हिमोस्टॅसिस, हा याला प्रायमरी हिमोस्टॅसिस. म्हणजे प्लेटलेटने त्याचे प्लग तयार करणे, तो प्लगने मग रक्त गोठवण्याच्या प्रक्रियेमध्ये मदत करणे, आणि मग त्याला रक्त गोठवून प्रिव्हेंटला विश्रांती देणे. So this happens in the steps of hemostasis. This is very simple mechanism. So next we will see in the next lecture. Till then we will stop our discussion here. Okay, see you in the next lecture.